All right, so I'm just gonna plug this thing in and starting to. All right, so yesterday I asked you guys to do this question along with watching some videos. So let's go over them. Let's go over these section right now. So explain the movement of ions in the neuron that has each membrane potential. So for A, minus 70 millivolt, that is your resting potential. So what's happening during resting potential? What's going on? Yes.
The resting potential is maintained by transport of the sodium to potassium pump, so sodium comes out and potassium.
Perfect. Thank you. Okay. Now, B and C, this one, considering the fact that B is minus 51, the other one's just minus 49, let's just say the threshold potential is minus 50. Okay. I know in the notes and textbooks say it's minus 50 to minus 55, but considering the question is like right next, almost the numbers are right next to each other. So let's just say threshold potential is minus 50. So what's happening at minus 51 then? Anyone? Yes?
It's past the threshold, so there's more voltage gated sodium channels that are open and sodium ions are rushing.
Do you remember this is on the negative axis, so minus 51 is actually lower than minus 49? Okay. So before threshold potential, what happens?
There's still voltage gated channels that are open, but there's not more that are open.
True. Some of those sodium voltage gate channel are open, so some sodium ion are rushing in, but not that much. Now minus 49 is past the threshold potential now. So more, if not all, the sodium voltage gate channel will open and sodium ion will rush in to the neuron. Okay, into the past the membrane, to the inside of the membrane. Are we good? Okay. So D, 0 millivolt. Now 0 millivolt, we on the action potential we have 0 millivolt twice. So let's just say the first time. Let's just talk about the first time the 0 millivolts on the way up. What is moving, and how, and where? So on its way up, that 0 millivolt, what is moving and where? Yes?
Sodium.
Yes, sodium is moving in, ion moving in, sodium gated channels are open. On the 0 millivolt, on the way down, so what is moving where now? Yes? Potassium ions are moving out. Excellent. Okay. Potassium voltage gate channels are open at that point, so potassium is moving out. Plus 30 millivolts, which bring us to the peak of the action potential or the overshoot, according to the video. So what's happening there? Yes?
It's the peak of action potential, and so the voltage gated sodium channels close, and the voltage gated potassium channels open, so potassium is rushing out.
Good. So sodium ion voltage gated channel closes, so no more sodium movement. Potassium ion channels start opening, so there's more starting potassium ion leaving the neuron. Okay? Questions? No? Okay. Okay, so if there's no more question. So we're looking at, so let's look at an overall what we have learned so far. So we have learned about the structure of neuron, and yesterday we talked about action potential, depolarization, and all that. Okay, so basically, we've been really talking about when the neuron is stimulated and a stimulation happens, and the nerve impulse travels down the axon. Now, if there's myelin sheath, it will go through what type of conduction? What do you like to put on fries? I know there could be many answers, but I'm looking for one as a hint. Not the one that I was hoping for, but saltatory conduction. Okay, if there's myelin sheath, saltatory conduction. If there's no myelin sheath, it will be continuous conduction. Okay, going down to the axon terminals now. So what happens? Now keep in mind, we are really talking about, trying to find a copy that I didn't really highly written on. We are really talking about at this point a number of, we're really talking about here. You have each neuron usually have a number of neurons connected to it and sending information, right? So how is the signal being sent from one neuron to another, or from one neuron to other cells, okay, like muscle cells, for example. So we're going to talk about that connection. So we're going to talk about something called synapse. We're going to talk about synapse and synaptic cleft. Basically, the connections between two neurons, okay, are called synapse. Remember, when you're learning something, when you're practicing something, essentially when you're building that muscle memory during practice, it could be music, it could be sports, it could even be washing dishes. When you're building that muscle memory, you're forming these connections. Your neurons are forming connections known as synapse. So at the synapse, we've been so far talking about electrical signals. Okay, nerve impulse is a signal, electrical signal. Now we have to convert it to chemical signal. That chemical signal is, we're going to use neurotransmitters. Okay. From the videos yesterday, you should have heard of acetylcholine. You should have heard of serotonin with the SSRI video. You should have heard of dopamine with the cocaine video. Okay. There are hundreds, probably more than hundreds now, because the textbook I was looking at yesterday was relatively old. So in this case, there are hundreds of chemicals which either we know for sure is chemical, it's neurotransmitter, or we suspect they are neurotransmitters. Okay. Now there are three parts to this section. So actually, you know what? I might have to draw on one of these new ones. Otherwise, it's hard to. Oops. Oh, here. Okay, so on these new ones, on this blank diagram. So what am I looking at? This top part here, that is the presynaptic cell or presynaptic neuron. Cell or neuron, okay? Hopefully you can tell that it's a neuron by context. We've been talking about neurons really much the entire time. And this gap in between the two neurons, that is the synaptic cleft. Basically, in biology, if you hear the word cleft, generally it's a gap, something. Like some kids are born with a cleft palate. So there's a gap right at their heart or soft palate, basically back on top of their mouth. Can be surgically repaired. Between two neurons. And this part here down below, that will be the postsynaptic cleft. Not postsynaptic cleft, postsynaptic cell or neuron. Okay. So right now, what we're talking about is the electrical or the nerve impulse have traveled down the axon onto axon terminus of the presynaptic neuron. So what happens now at this spot? You good? So we'll kind of set the stage. This is quite important. As I said, learning things requires requires forming these connections. Okay, so learning things, memory, the ability to do a lot of these things, the idea of neuroplasticity. Okay, all related to this. Okay, so let's go up there. Can you guys see on the back? Maybe I'll use a darker color. Hopefully it will be more clear and obvious. So synaptic transmission begins when the something reaches axon terminal buttons. It's N and I. What kind of word do you think they are? N something, I something reaches. Yep. Yes. Perfect. Thank you. Nerve impulses. Okay, nerve impulse. Nerve impulse reached the axon terminal button. Depolarization of the axon terminal membrane. So we're talking about this side. So we're talking about this side here. Causes voltage gated something ion channels to open and something ion rush into the terminal. So if you watch the video, hopefully that will remind you. Anyone? What kind of ion?
Potassium.
Pardon?
Potassium ion.
Hmm? Sorry, say again. Yes.
Uh,
Close, not quite. Yes? No. What? Calcium. How did you know? The video, good. Also, guys, if you read number 3, it's right there. Ca2 plus, Ca2 plus. Okay? Look, number 3, calcium ions. Triggers the release of, oh boy, neurotransmitter. Neurotransmitter, I'm going to zoom in. By causing the synaptic vesicle, VSICLE, closest to the synapse to fuse with the presynaptic membrane. Okay? So neurotransmitter and vesicle. Okay? How are we doing? So let's look back at the diagram underneath. Okay? So nerve impulse arriving, that's really step 1. Step 2, depolarization causes calcium ion to rush into the terminus. So here, these would be the calcium ions. And these, the lighter color one, would be the calcium ion channels. Calcium ion channels. And this here would be the neurotransmitter. And this would be the synaptic vesicle, SICLE. And that's number 3. These will fuse with the presynaptic membrane. How are we doing? Okay, number 4. When vesicle fuse with the presynaptic membrane, they empty their contents into the synaptic cleft. Okay, the neurotransmitter moves across the synaptic cleft and binds to the. What would it be? Binds to what? Is an R word. Oh, is an R word. Yes. Nope, not quite. Yes. Receptor. Thank you. I shouldn't have put the E part. The receptors on the postsynaptic membrane. Okay. Number 5, the response of the postsynaptic cell depends on the type of particular neurotransmitter. In most cases, the binding of the neurotransmitters results in a something opening and some ion enters postsynaptic cell. A hint, if you look at 6, this initiates action potential by moving down the postsynaptic neuron because now it's depolarized. So what do you think is opening? what kind of things opening, which if it is enough, can trigger an action potential? Yes. Good. Sodium ion channel. In some ways, that's why you shouldn't cut out salt from your diet, although you shouldn't eat that much salt in the first place. But salt is important. Okay. So let us go down here. You zoom back out a little bit. So number 4, you have neurotransmitters in the synaptic cleft. So I will say that about here, and some of them will be binding to neurotransmitters receptors, which is not shown, unfortunately. So these are receptors. These would be neurotransmitters. Okay. And number 5 and number 6 essentially, hopefully, will send another impulse down on the postsynaptic neuron. Okay, number 7. Neurotransmitter is degraded by something and is released from the receptor protein. This is enzyme. Okay, so those things don't get stuck there forever. They eventually have to come off. They will get broken down. And then the ion channel closes to the sodium ions. And then the neurotransmitter fragment diffuses back across the synaptic cleft to be reassembled in the terminal buttons of the presynaptic neurons. So here, now number 7 is not really shown. Number 7 not shown. But number 8 here, okay, number 8, some of these neurotransmitters will, after getting broken down, will get reabsorbed back in. Okay? That reuptake is important. That reuptake is important. Okay? Okay, questions? Yes? Phoenix, did you put up your hand? No? Okay, I wasn't sure. Okay, so now here's a specific example. Here's a specific example. And I believe this is also the example in the video. Acetylcholine. Acetylcholine is very important simply because It's one of those major neurotransmitter, especially in between the neuron and muscle fibers. Okay, so acetylcholine, okay, so neurotransmitters, many synapses. So it's made by combining choline, which is from diet, and an acetyl group during the aerobic respiration. Any of you guys remember which part of the aerobic respiration would produce an acetyl group? It's a very short section of it, of aerobic respiration. Yeah? Link reaction, good. Good. You have acetyl CoA, acetyl. So, anyways, now it is so acetylcholine is loaded into vesicles. So these are the vesicles with acetylcholine loaded into vesicles and released into synaptic cleft during transmission. Okay, so all these are in the synaptic cleft. Postsynaptic membrane has receptor which acetylcholine will bind and remain for a short period of time. Okay, so the postsynaptic membrane has receptors. Okay, will bind and remain for a short period of time. Uh, these receptors, as well as a neurotransmitter, are highly selective. They are highly selective, okay, or specific. I think it's specific. Selective. Selective is membrane. Specific. By the time after it binds, after a while, you will have this enzyme acetylcholine esterase. Acetylcholine esterase. Okay, it breaks down the acetylcholine. Okay, the choline is then reabsorbed and converted back into acetylcholine. So it's not shown here, but again, the choline will get reabsorbed. It is important for the neuron to get these neurotransmitters back. Okay. Now, yeah, I think this diagram was the same diagram that was drawn on the board a month or so ago, because you can actually have it, as you see here, connect to a muscle cell. Okay. Now, how can one of these things get messed up? These newer kinds of insecticides called neonicotinoids, okay, they are synthetic compounds very similar to nicotine, so neonicotinoids. It's used as insecticides. What they do is that they bind to the receptor site in the central nervous systems of insects. Now, in this case, acetylcholine esterase can't break them down. Okay? Can't break them down. So the binding is irreversible. So, what type of inhibition do you think this is? Going back to your unit 2. You're blocking the substrate by directly taking its spot. Yes. What? Competitive. Thank you. It's a kind of competitive inhibition. Okay? It's a kind of competitive inhibition. Okay? So now the receptor is blocked. Receptors are blocked. Synaptic transmission is prevented. Consequence is paralysis and death of the insect. Now, an advantage of using this type of insecticide, it's non-toxic to human and mammals. Okay, they they just don't bind to our uh, acetylcholine receptor as strongly as it would bind to an insect one. I don't know why. Probably some structural differences. And in some ways, this is important. How many of you have walked past someone's lawn, and on the lawn there's a sign saying, "Oh yeah, we just sprayed insecticide, so try not to walk on our lawn, or don't let your dog walk on our lawn." Like it is one of those things you have to watch out for if you spray insecticide. Or- Now the problem about this is the effects on honeybees and other beneficial insects. And apparently on honeybee, uh, not so much as paralyzed and deaf, but causes disorientation. So, in as you might notice, honeybees usually use the sun to kind of direct themselves to navigate, but these pesticides actually cause them to disorientate. So instead of flying back towards the hive, they'll fly way too far away. By the time they come to, they just can't make it because they're way too far. Okay. Prob How many of you guys heard of the problem with honeybee population collapsing? I think this is one of those possible essay question last year. Okay, causes collapse of honeybee population worldwide. Okay. Are you guys good? Now, last but not least, we're back down here, this diagram. Now, as you can see here, this neuron D, it gets information or it gets input from three different neurons, right? A, B, and C. So this part here, a typical postsynaptic neuron has synapse with many presynaptic neurons, okay? It may be necessary for several of these to release neurotransmitter at the same time for threshold potential to be reached and nerve impulse to be initiated. So let's pull up here. So if you look at this membrane potential graph that is on the neuron D, you might notice if neuron A fire its neurotransmitter, there's a little blurb, but it doesn't cause a depolarization. If neuron B fire its neurotransmitter, cause a little blurb up, it doesn't cause a action potential or depolarization. Okay. So from this we can tell A and B, they are stimulatory or excitatory. excitatory. I think that's how you spell it. I'll have to double check on that. Okay. So stimulatory or excitatory. I don't remember how to spell excitatory. So on their own, A and B, it probably won't cause enough of the depolarization for action potential occur. So in this case, you might need both of them. You might need both A and B to fire off their neurotransmitter, so D would actually cause an action potential. Okay. Release neurotransmitters together. Okay? So these two A neurons, A and B individually. Okay? A and B on their own doesn't cause an action potential or nerve impulse to happen. Now, any questions so far? So what's happening? What's this? What's the deal with C then? I'm gonna ask you guys then. So what's the deal with neuron C? Why is there like a dip down? Why do you think there's a dip down? So if neuron A and neuron B they stimulate or excites the other neuron, what does it look like neuron C is trying to do? Yes. Sorry, say again. Perfect. So it seems like neurons C, the neurotransmitter that they sent out, could be inhibitory. Inhibitory. Okay. A very well known inhibitory neurotransmitter is GABA. GABA. Okay. GABA is an inhibitory neurotransmitter. So not all neurotransmitters supposed to get things all fired up. Some are really just to calm everything down, so to speak. Okay. And there are so many of these scenarios that could happen. So, for example, I was talking with Miss Randall yesterday, and how many of you had taken grade 11 physics? Okay, good. So now, in situations like this, where A and B, both stimulatory, fire their neurotransmitter together, you have what's the term you guys you use when you have two waves that are in sync, combined together and forming a much bigger wave? I think it's something like construct. constructive interference, if that's what I remember. I don't remember correctly. So if you have two waves that are kind of firing at the same time and they're both constimulatory, you get a much bigger one. If you have, let's just say, one A and one C firing at the same time, then you might just have it kind of basically balance each other out. Destructive interference. You guys, yeah, okay. Last time I looked at this stuff was OAC physics, so I don't quite exactly remember the term. So in some ways, there's many possibilities, and this is one thing where, like, if we look at individual neurons, it fires impulses. But when you have a whole bunch of neurons, fire impulses sending different kind of neurotransmitters together, and some neurons trying to like figure out, so should I be firing? Should I not be firing? That creates all that together comes and create our nervous system. Okay, that allows us to write poetry, analyze idea. This is a bad idea. This is a horrible idea. All these things. Yes.
What happens then with, more specifically for like the relationship with the muscles? What happens when you like with involuntary movement, like the twitch or something?
I would say that's not my specialty. So in some ways, I can't give you like an answer. My educated guess, it would be if you have these kind of interference, it might be probably different level of muscle tension. Different level of muscle tension that will probably cause results in it. And if there is a neurotransmitter issue, then in this case, you can have either involuntary contraction. You can have paralysis and death. Like, for example, here, this acetylcholine receptor mechanism, if I remember correctly, some nerve gases works like that. It basically blocks this neurotransmitter happening, and people die in seconds. Okay. Does it help answer your question? Okay. Other questions? Now, what I want you guys to do is this. Where is it? So what I want you guys to do is the following two things. SSRI and cocaine. If you have not watched those videos, make sure you watch the videos. There's a video that I want you guys to watch. Okay. Transmitter. There's a video showing different 4 major neurotransmitters in mental health. It was done quite well, and you know what? I'll look at it and then I'll look for it specifically and then come back. It was a very I'll look for it and I'll come back and add the link. But there is a link here. There is a link here on the unit planner. Basically, this will take you to this website. Originally, it's in Dutch. Select the English or whatever language you prefer. I want you to pick 2 drugs of your choice on this website and look up how does it affect the synaptic transmission, because This is how the mechanism of action for a lot of drugs. Okay, I think someone, I don't remember in this class, were researching, their paper was on ecstasy. So here it is. Okay. Uh, yeah. So make sure you look it up. It's actually done quite well. Okay. Again, I do not endorse drug use in this course. Okay. Any questions? What you should be doing? No? All right.